Welcome back, everyone. Welcome to this last session of the day. I am Gregory Bedekaratz, professor in animal biosciences at the University of Guelph, and I will be moderating this last session. This last session focuses on some of the environmental management and as well a couple of presentation on layers. Before we get started, I would like to thank Egg Farmers of Canada for sponsoring this session. We all know how much they are involved in sponsoring all the research that has been presented so far today, and we are absolutely grateful. So without any further delay, I will introduce our first speaker of the session, Awais Ghaffar from the University of Calgary, who will be presenting on the impact of bioaerosols on the health of the poultry.
Okay, thank you so much for the introduction. Today I would like to talk about the impact of the barn bioaerosols on the health of the poultry. The barn bioaerosols include abiotic or biotic factors. Abiotic factors, they include different gases, for example, ammonia, hydrogen peroxide, and nitrate, and carbon dioxide. And biotic factors, they include different pathogens, for example, different viruses or bacteria or any other pathogens. A lot of gases, they are being produced within the poultry house by different mechanisms, and like the desirable range of those gases, it's okay, but sometimes due to different factors, their level increase in the environment. So like particularly in the poultry house, so they can go up to the lethal levels. So for every gas, there is a specific mechanism of the production, and there are specific desirable and lethal levels. A lot of bacteria, they are actually normal inhabitant of GIT tract of chickens. So from feces, they can come into the litter, and from litter, they can go into the environment. And in addition to those bacteria, there might be certain other bacteria. For example, if there is a problem which is being occurred at the farm, you can see certain pathogenic bacteria and even some other pathogens as well.
Got it?
Yes.
Okay. So what would be the consequences, those biotic and abiotic factors? Their additive effect, they may bring...
yeah, perfect.
So I would have access to this one, yeah?
Can you hear me?
So when they call your, or when they introduce you, then you...
Okay.
Yeah, go ahead. Sorry.
Oh, sorry. Okay. So those biotic and abiotic factors, their interactive effects, they can bring some consequences to the birds and the birds handlers. For example, the bacteria and antimicrobial resistance genes, particularly, they can have public health significance. You can find some antimicrobial resistance genes of public health significance. And about gases, particularly ammonia, its higher level, if it is present in the environment, particularly in the house, due to any problem, then it can cause problems to the bird handlers as well. So keeping in mind all of those backgrounds, we designed this study, and the objectives of our study are: the first objective is to characterize the level of airborne biotic and abiotic factors in layered chicken barn relevant to the Canadian climate conditions. Here I would like to mention that I'm particularly talking about Canada. We could not find any study focusing those particularly bacteria and antimicrobial resistance genes in the environment of poultry in Canada. So here comes the methodology. We will go for sampling in Alberta, and we will select 10% of the farms. There are a total of 176 farms, so we will go to 10%, 18 barns, including 3 free range, 4 free runs, 5 enriched, and 6 conventional forms. And in order to compare different seasons, we will go in the summer as well as in the winter, in order to like compare the season as well as in order to compare different type of the farming practices. And we will collect we have designed a questionnaire. We will collect all the flock health information in that particular questionnaire. So for gases, we will use different portable devices, for example, for carbon dioxide, hydrogen sulfide, ammonia nitrate, and we will also measure the body weight, temperature, humidity, and different other factors at the farm. So here is the machine which we will use. Actually, it is the air sampling machine, XMX. We will use it at 5 different locations within the barn. And we will collect we will extract the DNA from the samples, and we will go for the metagenomic sequences. So now what is the metagenomics? It will tell us the genetic makeup of the particular sample. As we are focusing on the air samples, so we will came to know about the antimicrobial resistance genes and bacterial taxonomy in our samples. And like by different bioinformatic softwares, we will analyze our data. The objective 2 is to determine the effect of barn air quality on the development of vaccine-mediated immune response in layer chickens. In order to fulfill this objective, we will select 10 to 20 birds per barn according to the type of the barn, and we will evaluate the serological response of Newcastle disease and infectious bronchitis. We will go for sampling before and after vaccines. Here is the project team. My supervisor, Professor Dr. Faisal Abdul Kareem, who is the PI of this project, and he is expert in immunology. And then Dr. Karen and Sylvia, they are expert in AMR and bacteriology, and Dr. Hansi is expert in chemistry. So this project will actually give us the baseline data of different biotic and abiotic constituents of poultry houses. Because I have told you this is the first time we are going to do it, so it will tell us about the baseline data. After that, it will be a baseline study for many other different research projects. And if found something bad, we can also optimize the management strategies according to the results which we will find. I would like to acknowledge my supervisory committee, my lab members, and our funding agencies. And thank you so much. If you have any questions, you're most welcome.
Thank you very much for a great presentation, Awais. If you have any question, please use the Q&A chat there so I can communicate them to the presenters. Now, I've got a quick question here. When you listed the type of barns or the types of farms that you're going to be using, do you have a kind of a standard set for the barn design? What I mean is you could have like conventional group, but there can be multiple different types of barn design in conventional, for example, multi-tier system, or if there's only one floor, so on. So are you selecting those farms so that you've got a homogeneous group for each?
Oh, thank you. Great question. Like we will be going to different type of the farmings. We like they're already defined, like which one is, we will define that these are the free runs farm, these are the enriched farm, these are the conventional farm, and accordingly we will visit them. And because of the number of the farms, we have selected that particular number in our sampling strategy.
Yeah, no, and I think that may be a good point, especially some may have different ventilation system or different heating system that could compound the results to a certain extent.
Exactly. At every farm, definitely there is different type of management system. If there is more litter, for example, at a farm, there is more production of gases because of the decomposition and things like that, and maybe we will find different type of bacteria. So the idea is to compare the gases and bacterial count not only in different season but also at different type of the farms.
All right. Well, thank you. Thank you very much.
Oh, thank you so much.
I